When the pharmaceutical industry discusses digital transformation, the conversation usually defaults to hardware. Lab managers are often told they need a $250,000 automated plate reader or a fully robotic incubator to modernize their microbiology quality control, or QC, programs. But bringing heavy machinery into a regulated space requires months of installation, operational, and performance qualification. These validation protocols and test scripts consume hundreds of labor hours before a single actual sample can be processed in a compliant manner. This flowchart shows the real constraint in a QC lab. While analysts can streak plates and read results quickly, the pipeline gets clogged at the desk. Microbiologists are trapped executing cognitive and regulatory processing tasks, like writing investigations, researching organisms, and reviewing standard operating procedures. Generative AI clears these text-heavy choke points from your existing desktop, removing the need for new hardware validation entirely. Large language models handle the heavy lifting of research and documentation that keep staff stuck at desks until 7 p.m. This software-first strategy functions as a low capital expenditure solution. You achieve a high return on investment by optimizing the logic of your existing operations, circumventing the need for massive lab budget approvals. Building efficiency into the lab's logical workflows provides the gains of automation without the capital expense of a $200,000 automated incubator. This approach frees microbiologists from their desks to focus on actual laboratory science. This diagram illustrates the lab stack. To improve lab performance, we begin at this foundational layer, daily organism research and identification, where analysts spend hours establishing data context for their findings. In non-sterile manufacturing, analysts must determine if a recovered microbe is an objectionable microorganism, a species that poses a risk to the specific product or patient. Traditionally, this requires hours of manual research across Google Scholar or physical copies of Burgess' Manual of Determinative Bacteriology. An AI model operates differently by simultaneously cross-referencing external databases, historical recall data, and FDA guidance documents. It merges these streams into a single, cohesive risk evaluation. A specific prompt can evaluate the Burkholderia cepacia complex within the context of a high-water activity nasal spray. High-water activity refers to a product's moisture content that is readily available for microbial growth. The AI parses the pathogenicity and regulatory history of that specific formulation in seconds. Automating the research phase shifts the microbiologist's role from manual data retrieval to high-level risk evaluation. The next layers of the operational stack involve standard operating procedures, or SOPs, and analyst training. The standard read and understand training model often fails to prove actual analyst competency. This creates a compliance vulnerability because the signature only confirms the document was seen, not that the procedure was mastered. AI can ingest a complex microbial limits test SOP and generate a challenging, tiered quiz. By focusing on incubation parameters and out-of-specification, or OOS, handling, the system creates a tiered training program that tests the ability to handle deviations. An OOS is any test result that falls outside of established acceptance criteria. This document parsing graphic demonstrates how AI functions as a regulatory filter. Before an environmental monitoring procedure is routed for approval, the AI identifies ambiguous phrases like check periodically and suggests rigid, auditor-friendly language like at a defined frequency. Enforcing active competency and scanning for ambiguity provides a proactive shield against FDA 483 audit observations. A 483 is a formal notice issued by the FDA citing potential regulatory violations observed during an inspection. Layer 4 addresses data trending. Many labs manage environmental monitoring data in manual spreadsheets because they lack the budget for a comprehensive laboratory information management system, or LIMS. Raw, unstructured excursion data contains systemic patterns that are difficult to see in a spreadsheet. While a single recovery might look isolated, the aggregate data often reveals an underlying trend. This scatterplot shows dozens of raw data points. AI can cluster these points to identify correlations, such as a specific Staphylococcus species appearing only on days when a specific analyst is performing the sampling. The model can also detect a slow 3-month upward trajectory in microbial recoveries within water for injection, or WFI, loops. This emergent pattern is the biofilm signal. It indicates a systemic contamination drift long before a human reviewer would flag a single isolated excursion. Identifying these drifts early shifts the lab from reactive troubleshooting to predictive maintenance, preventing major contamination events before they occur. The apex of the lab stack involves macro-level regulatory synthesis, specifically annual product quality reviews, or APQRs. Generating these reports is a significant administrative burden. Managers must manually categorize a year's worth of lab investigations and deviations to fulfill regulatory requirements. This diagram illustrates the acceleration of that process. AI ingests hundreds of deviation files and categorizes them by root cause. This immediately reveals, for example, that 40% of deviations are caused by sampling errors. The AI then drafts a structured corrective and preventative action document, or CAPA. This CAPA suggests specific retraining based on the identified root causes, turning data entry into actionable strategy. This synthesis compresses a week of manual data categorization into 30 minutes of strategic analysis. Using AI in a regulated space requires strict adherence to good practice regulations, or GXP. This environment demands rigorous validation of any process that generates official records. The Lawhon Doctrine addresses this. In pharma quality control, AI is a process generator, not a biological search engine. This infographic shows the risk of unvalidated use. Copying AI outputs directly into official records is an immediate regulatory violation. The system provides a draft, not a verified fact. The mandatory solution is visible in this flowchart. You must implement a human-in-the-loop architecture, where every AI output passes through a rigid gate, requiring a human review and signature before it enters the official quality management system. Compliant labs use a GXP AI Audit Defense Kit to record and store every interaction session. These logs ensure that the entire logic path of the AI is auditable and stored as a GXP record. Eventually, this logic can be embedded directly into an electronic quality management system, or EQMS, like AmpleLogic. This automates the validation choke point within your existing workflow. By securing the validation choke point, microbiologists move away from spreadsheet entry and return to science. The result is a lean, smart laboratory, achieved without a single piece of new hardware.